At this ASCO GU Symposium 2022, we showed very exciting data on antibody drug conjugates in urothelial carcinoma. In the metastatic platinum-refractory setting, we showed data from the Trophy U01 Cohort 3 with pembrolizumab plus Sacituzumab govitecan. We also showed data from the interesting combination of nivolumab plus Trastuzumab deruxtecan in platinum-refractory disease as well. And we also showed data in the neoadjuvant setting with single-agent Enfortumab vedotin in cisplatin-ineligible patients undergoing radical cystectomy. We're very excited at the ASCO GU Symposium 2022. We had the opportunity to present data along with my co-authors and co-investigators of the Trophy U01 study, Cohort 3, Sacituzumab govitecan plus pembrolizumab in the platinum-refractory setting of advanced urothelial cancer. This study evaluated the combination of an antibody-drug conjugate, Sacituzumab govitecan, we'll call it SG, which is targeting TROP2 in the membrane of urothelial cancer cells, linked to SN38, a metabolite of irinotecan. And this antibody-drug conjugate was combined with pembrolizumab, an anti-PD-1 agent. These patients had previous platinum-based chemotherapy, half of them in the perioperative setting and the other half in the first-line setting of advanced urothelial cancer. We had 41 patients enrolled in this study, and 68% of patients had visceral metastasis, 29% had liver metastasis. The median time between the end of prior chemotherapy and the trial screening was only 1.6 months, showing that these patients had rapid progression, and the response rate after platinum-based chemotherapy was only 15% in those who received it in the first-line setting. Despite these negative prognostic factors, the objective response rate was 34%, and the clinical benefit rate was 61%. With a median follow-up of 6 months, the median PFS was 5.5 months. Toxicity was as expected with each agent by itself, pembrolizumab, anti-PD-1, and SG, antibody-drug conjugate against TROP2. We had neutropenia, anemia, diarrhea, fatigue as common side effects, and the treatment-related grade 3-4 adverse event happened in about 59% of patients. 4 out of 41 patients had immune-related adverse event requiring oral steroids. Overall, the combination was feasible with a manageable safety profile. The data are not practice-changing, but definitely support further evaluation of this combination in the second-line setting, but maybe in the first-line setting of advanced urothelial cancer. We saw some very interesting data from a clinical study by Dr. Galsky and colleagues evaluating Trastuzumab deruxtecan, an antibody-drug conjugate against HER2. This drug conjugate has been tested in DESTINY-04 trial in breast cancer with impressive data and links this anti-HER2 antibody through a topoisomerase 1 inhibitor payload. This antibody-drug conjugate was combined with nivolumab, an anti-PD-1 agent, in patients with platinum-refractory advanced urothelial cancer. There were 2 cohorts, Cohort 3 with high IHC +2+3 expression of HER2, and Cohort 4 with IHC +1 low expression of HER2. The combination of the Trastuzumab deruxtecan and nivolumab showed a promising overall response rate by independent review committee. It was 37% with a median duration of response 13 months, median PFS of 7 months, and median time to response 2 months. Median overall survival was 11 months. This was a single-arm, not randomized study. Grade 3 treatment emergent of any cause adverse events happened in about 3/4 of the patients, and the most common any grade treatment emergent adverse events included nausea, fatigue, and vomiting. The drug-related interstitial lung disease, pneumonitis, occurred in about 1/4 of the patients. Overall, the combination of Trastuzumab deruxtecan and nivolumab appeared promising. The data are not practice-changing, but definitely support further evaluation of this combination in advanced urothelial cancer. We showed data from the EV-103 phase 1b trial in the neoadjuvant setting before radical cystectomy in cisplatin-ineligible patients. This study, this particular cohort H, presented by Dr. Petrilak, included 22 patients, mostly with clinical T2 disease, who received neoadjuvant Enfortumab vedotin on days 1 and 8 on a 21-day cycle. These patients received 3 cycles of Enfortumab vedotin and then underwent radical cystectomy, lymph node dissection. From the 22 patients, we had 3 patients who died after surgery, and this requires further attention. Overall, pathologic complete response rate was 36%, and the pathologic downstaging was about 50%. The toxicity profile was as expected, with no new safety signals. The most common treatment-related adverse events include fatigue, alopecia, and taste changes. As I mentioned, this is an important study, but it's not practice-changing, but definitely support further evaluation of Enfortumab vedotin in the neoadjuvant setting in 2 trials that are phase 3, the Keynote B15 trial in cisplatin-ineligible patients and the Keynote 905 trial in cisplatin-ineligible patients. In both those trials, the combination of pembrolizumab plus Enfortumab vedotin is being evaluated, and we look forward to those trials in the future. So interesting data we saw from cohort 8 from EV103, not practice changing yet, but supporting the ongoing phase 3 trials.